In this video, we will review the regulation of mean arterial pressure. Mean arterial pressure, like its name suggests, is the average pressure inside of the systemic arteries, and it's one measure of blood pressure used clinically. Mean arterial pressure can be calculated from cardiac output and total peripheral resistance. In other words, blood pressure is regulated by cardiac function and by blood flow through arteries. For this roadmap, we're going to start from this equation and work backwards. Recall from the heart unit that cardiac output is the rate at which blood leaves the heart, expressed as a volume over time. To put this into more familiar terms, cardiac output is equal to stroke volume, which is the amount of blood ejected from a ventricle, times heart rate. To break this down even further, we could say that stroke volume is determined by contractility, or the force of contraction generated in contractile cells, and heart rate is determined by action potential frequency in pacemaker cells. Recall that the heart can be regulated intrinsically according to the Frank-Starling law. The more blood that is returned to the heart, the larger the end diastolic volume and pressure inside of the ventricle. Because cardiac muscle is not at its optimal length at rest, when the cells become stretched, they maximize the length-tension relationship in their sarcomeres and can generate more force. We could summarize this by saying that more blood being returned to the heart results in greater stroke volume and greater cardiac output. The heart is also regulated extrinsically by hormones in the autonomic nervous system. Recall that both contractile and pacemaker cells are innervated by the sympathetic nervous system. When the cardioacceleratory center and cardiac nerve are stimulated, epinephrine and norepinephrine released onto beta-adrenergic receptors on contractile cells ultimately increases cytoplasmic calcium and leads to an increased contractility and stroke volume. When these same receptors are stimulated on pacemaker cells, action potential frequency and heart rate increase. Relating this back to the equation at the bottom, we could say that sympathetic stimulation increases stroke volume, increases heart rate, increases cardiac output, and therefore increases mean arterial pressure. Activation of the cardioinhibitory center and vagus nerve, on the other hand, decreases mean arterial pressure, but to a much lesser degree. This is because only pacemaker cells are innervated parasympathetically. When acetylcholine is released onto muscarinic receptors on pacemaker cells, action potential frequency and heart rate decrease. This decrease in heart rate decreases cardiac output and therefore decreases mean arterial pressure. All of this is to say that cardiac function has a significant impact on blood pressure. However, according to the equation at the bottom, total peripheral resistance is also a contributing factor. One option to increase total peripheral resistance is to increase the viscosity of the blood by increasing hematocrit through the process of erythropoiesis. However, this is generally not a consideration in short-term blood pressure regulation because these changes are slow to develop. However, since resistance opposes blood flow, it does explain why polycythemic individuals carry an increased risk for stroke. The more important regulator of total peripheral resistance is arterial radius in response to hormone and autonomic inputs. Like contractile cells of the heart, these innervations are primarily sympathetic. Stimulation of alpha-adrenergic receptors results in contraction of smooth muscle cells in the tunica media and narrows the radius of arterioles. This process is known as vasoconstriction and increases total peripheral resistance and therefore blood pressure. Stimulation of beta-adrenergic receptors results in the relaxation of smooth muscle and increases the radius of the arterioles. This process is known as vasodilation and decreases total peripheral resistance. Generally speaking, hormones are more important in mediating slow and long-term changes in blood pressure and go beyond the scope of this video. However, atrial natriuretic peptide released from the heart is a vasodilator, and angiotensin is a vasoconstrictor. The last piece of this puzzle is the veins, which regulate the amount of blood being returned to the heart. Sympathetic stimulation of veins decreases venous compliance, which allows for more efficient return of blood to the heart. This increases end diastolic volume, stroke volume, cardiac output, and mean arterial pressure. Keeping these mechanisms in mind will help you to better predict how changes in baro and chemoreceptor activity can rapidly adjust and compensate for small fluctuations in blood pressure and blood gases.